Okay, so we've determined that Ungerleider und Mischkin did two types of gave their test monkeys two types of problems: object discrimination and landmark discrimination. All right. So once one of these monkeys were trained, right, they used ablation to remove part of the parietal lobe and in half the monkeys, and then part of the temporal lobe was removed from the other half. So the yeah, so right here, this is the temporal lobe in the monkey brain, and this is the parietal lobe area right here. Okay. So after the monkeys were trained, they had either their parietal lobe or temporal lobe removed, not both, and then they were retested. And it turns out that if the monkey had their temporal lobe removed, right, as in here, they had problems with the object discrimination task, right? So the what pathway, okay? I know your slide here says where. That should say what. That's a misprint. um, but this would be the this has a this tells us it's discrimination with the what pathway. Whoops. Oh, darn it. There we go. Yeah. So it says what here, right? So discrimination with the what. Is it this object or this object? So what? Okay. However, if they had their tissue removed here from the parietal lobe, they would have discrimination problems with the where pathway. In other words, they were able to determine which food cup the food was in based on where this little marker was. They lost that ability. They after the ablation, they lost the ability to determine the relationship between this marker and which food cup had food in it. Okay, so what we have here is this occipital lobe, right? This is where the main visual processing starts, right? And like I said, we have two separate pathways: the dorsal pathway, which runs along the top here, and the ventral pathway, which runs along the bottom. So whenever you see dorsal and ventral in anatomy, dorsal means along the top and ventral means along the bottom. Okay, that's what those two terms mean. So the dorsal pathway runs up to the parietal lobe area, to the entire parietal region, and it has to do with where and how. So where is something, right, and how do you use it, right? How does it relate to my goals, right? Whereas the ventral pathway, which is the temporal lobe pathway, relates to what, like what is it? What is it called? Okay. The really fascinating thing here, the take-home lesson, is that we were able to separate out these two pathways, right? Where and how, and what. And we're going to find later that humans have the same pathways. Um, the what pathway is also called the ventral pathway, right? And the where pathway is also called the dorsal pathway. Okay. Now both of these pathways begin in the retina. Okay, they both start in the retina with those receptive fields, go through the ganglion cells, go through the lateral geniculate nucleus, and then go to the primary visual cortex, and then they branch out from the primary visual cortex, and then they separate out. Now, of course, when our brains are normally working, these two pathways talk to each other, right? So, if I see a hammer and I know where it is, right? So I know the where. I also need to know the how, right? So, and I can describe to you using language um, the where and the how and the why of hammers, right? And that's because those two pathways are connected to each other, right? I can access the where and the how and the why all at the same time because there are some interconnections, right? And of course, they do receive feedback from even higher brain areas. So, for example, we're going to learn that a lot of the things that when we see something that we can do something with, like pick it up or hold it, like a like a handle on a cup, for example, that involves processing with higher brain areas. Okay. The where pathway may actually be a how pathway or an action pathway. In other words, not only does it seem to tell us where something is, but it also seems to give us some information about how to use something. Okay. Um, it seems to have the dorsal stream shows functioning for both location and for action, right? Um, and we know this because there are people who have a disconnection between where something is, I'm sorry, what something is, and how to use it, right? So, for example, um, we have this patient here, and this is from Table 4.1, right, in your book on page 85, right? So we have this patient Alice. She has temporal lobe damage. If you remember, the temporal lobe is associated with the lower or ventral stream, right? Now, Alice, she just has temporal lobe damage, and this may be, for example, from a stroke. So Alice cannot name objects, so she doesn't know what things are. But she can tell you where they're located, right? So Alice might see, um, this card. Here's a card, Alice, right? And what is that? And she say, well, I don't know what it is, right? But she would be able to determine, she would be able to describe very accurately where it's located in space. Bert, on the other hand, might have parietal lobe damage, that the dorsal stream or the upper stream, right? He would be able to name objects, right? He would be able to say that's a card, right? But he would not be able to determine where that object is, right? He would not be able to say that object is close to this object, or uh, that object is to my left, or that object is to my right, right? He wouldn't be able to do that. Okay, so uh, there's a lot of evidence from human studies about these what and how pathways, right? So we have this patient, this very famous patient, named who went by the initials DF, right? These patients their anonymity was always protected, right? And this person had unfortunately suffered a brain damage from a gas leak. So apparently they were asphyxiated in a gas leak, and while their brain was not getting oxygen, they suffered brain damage. And the part of the brain damage they suffered was in their temporal area. So this ventral pathway was destroyed. And there was a very interesting phenomenon that was noted in DF, right? So basically, we had this the task where they tested him was this card task, right? Where basically you take a card. It's like sending a letter in an old fashioned mailbox, right? You take a card and then you put it in a slot like that, right? And if the slot's oriented like this, it's going to be very easy for you to pick the card up and put it in there, right? If the slot's oriented like that, very easy again for you to pick the card up and do that. Also, if I were to show you a picture of the slot oriented like that or like that, you could tell me. You could show me a picture of the card that would go into the slot. I would show you a picture of a card that's oriented like this or like this, and you could say, yeah, the card that's oriented like that would go into that kind of slot. Well, the really interesting thing is if you ask DF what kind of card goes into that kind of slot, this card or this card, he couldn't tell you. He did not know. He was not able to do it. That he could not do that task. But if you actually gave him a card and showed him the slot, he could get the card and insert it into the slot. He could do it, he just couldn't describe it, which is a beautiful separation of the what and the how components, the ventral and the dorsal pathways. Other patients would show the opposite effects, right? So if they had damage to their dorsal pathway, they would not be able to do the task. They couldn't do it, but they could tell you which card goes into which slot. And here's a picture of some of the data, right? Whenever we ask DF um, how a particular card should go in, if the card is if the slot is a vertical slot, if we were to ask him to take many guesses on how that card goes in, he would be all over the place, right? He'd be like guessing all over the place, whereas a normal person, a control person, would be pretty, pretty accurate in determining it. Yeah, that card goes in horizontally. But when you ask DF to put the card in, he does great, just all as well as a control group. Gunnell did a really interesting experiment where he it was a simple experiment, but it was pretty neat. Basically, we have two lines here, and the subjects were asked to look at this stimulus, right? And line one was actually a little bit longer than line two, right? In reality, but they were asked to look at the stimulus, but the numbers weren't there. It was just this thing and the lines, right? And they were asked first to kind of just use their fingers to guess how long each line was, right? So and they were just told, okay, hold your fingers apart. Don't just don't touch anything there. Just hold your fingers apart and tell me how big you think each line is, right? And they had little sensors on their fingers that would measure how far apart, how many millimeters apart their fingers were. So you had one group do that, and that was just the group that was here indicated here, length estimations. Then he had another group that was told, okay, you can actually grab the stimulus. You can actually go and you can put your fingers on it and use them to measure the length, right? And sure enough, what they found was that when people are able to grasp, when they're able to actually put their fingers up and estimate and use that to estimate the line length, they did really well. They were able to differentiate the very fine difference between the long line and the short line. But when they were not actually doing it, when they were not actually touching the line and figuring out and estimating its length by touching it, they did very poorly. As a matter of fact, they pretty much fell for this illusion here, which shows that line 2 is longer than line 1, when in fact line 1 is actually longer than line 2. One of the things that we've discovered in recent years, in human and in animal brains, is this idea of a module. Okay, a module being a part of the brain that processes information about a specific stimulus. Okay, so one of the reasons this idea has been talked about is because of research that has been done in monkeys and in people that shows that we have specific areas of our brain that process faces, right? So roles, this research mentioned here in the slide, roles measured responses in neurons in a part of the brain called the inferotemporal cortex, the IT cortex in monkeys. And he found that in monkeys, this part of the brain responds powerfully to faces, to other monkey faces particularly, and doesn't respond to other stimuli like stars and squares and circles and things like that. Uh, Human beings with a particular type of temporal lobe damage, which is the same general area as this in monkeys, can suffer a disorder called prosopagnosia, which is basically the inability to recognize faces, which is really interesting. So you can see the face, you just you cannot connect who it is or who you're talking to, which is really amazing. That we can perceive the face. I can tell you where the eyes and the nose and the mouth are, but we would have no ability whatsoever to recognize that as a face or recognize who it is. Right. So here we have this image of neurons that are firing for very particular stimuli. Right. Here we have a paddle with a stick attached to it, which causes great firing here. Then we have just the stick by itself. After we measure this, we find this neuron fires very powerfully to this circle attached to the stick. This particular neuron does not fire very strongly to this stick. It fires a little strongly to the paddle by itself without the stick, and it fires fairly strongly to a square paddle attached to a stick. The take-home message here is that this neuron, and this is recording from one neuron in the interotemporal cortex of macaque monkeys. This one neuron is unusually sensitive, or is very sensitive to just this type of stimulus here. Basically, this complex stimulus that involves a large area with an appendage on it.